So I'm glad that Nora agreed to present her data today. And Nora is an associate professor in the neurology department, and we were both also faculty on this BENFRA grant, which was investigating how botanicals can protect from age-related phenotypes, especially cognitive decline. And so I was involved in that using Drosophila, and Nora was involved in that using mice. And so she will tell us about that and some other data she has with these botanicals. So over to you.
Thanks, Doris. And thank you for the invitation. It's always fun to share our work with new audiences. Like Doris said, I'm going to be presenting some of the data that came out of this BENFRA Botanical Research Center that Doris and I were both a part of. I am going to be focusing on the mouse work that came out of my lab and kind of how that contributed to a clinical trial that actually just wrapped up using this same botanical agent that we were studying in this center. Okay, so if you're not familiar with it, the BENFRA Research Center was a U19 center here at OHSU, and it stands for Botanicals Enhancing Neurological and Functional Resilience in Aging. And basically, it's designed as this pipeline to kind of move botanical agents through rigorous preclinical testing to prepare them for effective clinical trials eventually. And it was a massive collaborative effort across a number of different labs here at OHSU, but also at Oregon State University, a few other institutions around the country. We even had some international collaborators and some companies that helped us source our plant material. So it was really a big effort. And the director of the BENFRA Center is Amala Saumianis, who did a great job of leading the whole endeavor. But the way that it's designed is to have these collaborative projects that can really start with a plant and start with chemists working on it to kind of prepare different extracts of the plant and use their methodology to characterize the chemical composition. And then working with labs like Doris's, using her Drosophila models, or other labs using cell culture, you could kind of go back and forth between the chemists and these biological model systems that are pretty high throughput and try to identify which botanical agents are showing the most promising effects, the endpoints that are related to age-related changes in cognition, sleep, and anxiety and depression. The goal of that intermediate step there is really to try to identify what compounds within the extract are most important for the different biological effects, and figure out what preparations of the botanical have high levels of those compounds, so that we are kind of like narrowing it down to a preparation that will eventually get used in a clinical trial. So after Doris and others work to figure out which are the most relevant compounds and which preparations are the most effective, it then moved on to the step where my lab was really involved, which was testing in mouse models to look at efficacy and also to identify translationally relevant biomarkers. So in this stage, we were looking at how to optimize route of administration and dosing parameters, but always with a focus on what would be most relevant in an eventual clinical trial. Because the ultimate goal of BENFRA is to, at the end of this pipeline, have a preparation that comes out that we feel really confident we know the right the way of extracting the plant material, what are the most important compounds to standardize it to in our preparation, how is it likely working, and what should we be monitoring in a clinical trial to gauge or to assess target engagement. So that's the big picture goal of the BENFRA Center. Like I said, it was really focused on looking at plants that could enhance changes in cognition, sleep, and mood that happen with aging, But for my talk today, I'm really going to focus on the cognitive enhancing effects of one of the plants that we were looking at, because it's a really interesting story, because it eventually led to this clinical trial that we just finished a couple of months ago. So as I'm sure many of you, if not everybody, knows, cognitive impairment is very common during aging. This image here, let me see if I can get the laser pointer. Arrow options. Nope. Oh well. Okay. Oh, here it is. Pointer options. Sorry about that. So in this panel over here, you can see different domains of cognitive function, and what you appreciate is that most of them are declining with age, although at different rates. And it's estimated that as many as 2 out of 3 adults have some level, older adults have some level of cognitive impairment. And the effects of aging on cognitive impairment are really even more pronounced when you look at specific conditions of cognitive impairment. And that's what this figure here on the right is showing. This is in people with mild cognitive impairment. You can see that the prevalence goes way up with age. So the plant that I'm going to talk about today is one of the main ones that we were studying in the Benford Center. It's called Centella asiatica. It's also marketed in the United States as the dietary supplement Go-To Cola. So you might see it that way, like at a natural store or something like that in the US. But it has a really long history of traditional use in Ayurvedic medicine as well as traditional Chinese medicine. And there are a variety of indications that it was used for, or it still is used for, in those traditional systems, but a number of them are related to improving cognitive function. And if you go to the literature, there's quite a bit available that's been reported in the preclinical space, confirming these cognitive enhancing effects and also showing effects on antioxidant and anti-inflammatory pathways and just evidence of neuroprotection in general. There have even been a couple of small-scale clinical trials that have been done using this botanical, and they have also supported the cognitive enhancing effects of the plant in both cognitively normal and cognitively impaired adults, as well as mentally impaired children. They have seen that Centella asiatica can improve cognitive function, but it's important to note for these trials that they all suffer from pretty significant methodological flaws that really limit how much you can interpret their data. Like most of them are really underpowered. Several of them were not placebo controlled. So that's all to say that while it is encouraging to see that some of the same effects that have been seen in traditional use and in the preclinical space are also showing up in clinical trials, we still don't have a definitive, robust efficacy trial to really hang our head on for, yes, this is an effective plant for cognitive impairment. And that's kind of the goal of what we were working on in the Benfra, is to get a preparation ready that we can then do just that kind of efficacy trial. So like all plants, essentially, Centella asiatica is chemically very diverse. This is just a little tree here that some of the chemists on the Benford team put together. And I'm not going to go through this in any detail. This is just to kind of give a sense of the breadth of chemical variety that exists in this plant, because that's a challenge with working with botanicals, that you can see a lot of variability in the levels of these chemical constituents depending on growing conditions, like the soil composition, the temperature, the rainfall. All of those things can dramatically affect these levels of these different compounds. So it's really critical when you're trying to create a standard preparation to use in a human trial that you know what the most important compounds are, so that you can try to standardize your preparation to have consistent high levels of those important compounds. So in the Benford Center, one of the first obvious questions we asked when we started working with this is, can Centella asiatica improve cognition in aged mice? And specifically, we were working with a water extract of the plant, which we call CAW. It's basically made like you would make a tea in the traditional preparation, where you just boil the dried plant material in water and then lyophilize that down to a powder, which can then be resuspended in the drinking water of the mice to give them exposure to the extract. So the way that this experiment worked is we had 18-month-old 67 black 6 mice that were treated with 10 grams per liter of CAW in their drinking water. And we compared them to 18-month-old mice that didn't receive any CAW and also younger 3-month-old mice that didn't receive any CAW. And they were treated for 5 weeks. In the final 2 weeks, they underwent behavioral testing to evaluate cognitive function. And we actually ended up seeing some pretty impressive results. This test here is called the Odor Discrimination Reversal Learning Test, and it is a two-part test that really lets you look at both learning and also the cognitive flexibility domain of executive function. So the way the test works is you train a mouse to dig in a bowl for a food reward. And in the first part of the test, the mouse is presented with 2 bowls, each containing a different digging material and with a different odor associated with it. So in this example over here, the 2 digging materials are plastic beads and sand, and the 2 odors are mint and vanilla. And in the first part of the test, which is called the acquisition phase, the food reward is always paired with the odor. So it would always be paired with the mint odor, regardless of whether it was the beads or the sand that smelled like mint. Because in every trial, one of the bowls smells like mint and one smells like vanilla, and one bowl has beads and the other has sand. But it randomly alternates how those digging materials and odors are paired. But in the first phase, the mouse has to learn that whichever one smells like mint has the food reward, regardless of digging material. And you just stand there and you count how many trials it takes the mouse to learn this. We define learning as 8 out of 10 correct trials in any bout of 10. And what you can see here in this graph on the left is that the older mice take many more trials just to learn this association than the younger mice. But the older mice that were treated with CAW have a dramatic improvement in their performance, and they're back at levels that are very comparable to these younger animals. As soon as the mouse hits that threshold for learning this first association, that immediately starts the second phase of the test, which is called the shift phase, where now it's the digging material that dictates where the food reward is, irrespective of odor. So in this part of the test, it would always be the beads that have the food reward, and it doesn't matter if they smell like mint or vanilla. And so the mouse has to realize that the rules have changed and start paying attention to this new discriminatory dimension of the digging material in order to learn this new part of the test. And it's that part, that ability to recognize that the rules have shifted, that really gets at the cognitive flexibility part of executive function. And if you look at this data here on the right, you can see that we're seeing a very similar pattern as we saw in the earlier just classical learning phase, where the older animals take many more trials to learn the new rules here. And the ones that are treated with CAW are behaving much more similar to the young animals, that there's a dramatic improvement in their performance in this task as well. And I'm just noting here that we've also done this test in our mouse models of Alzheimer's disease, and we see very similar improvements with the CAW treatment, that you get improved learning and improved cognitive flexibility in those mice as well. The other test we did in these older mice is the novel object recognition test, which assesses memory, specifically recognition memory. And it's a pretty straightforward test. Most of you are probably familiar with it, but if you're not, the way that it works is you put a mouse in an arena and you present them with 2 identical objects for several training sessions. Then at the end of the last training session, there's a time delay. Then you put the mouse back in the arena for a testing session where one of the objects is the same as has always been there during training, and the other object is brand new. The mouse has never seen it before. And because rodents are inherently exploratory, if the mouse remembers the object from training, it'll spend more time with the novel object. And that is what you're seeing here. If you look at these young animals, they're spending about 60–75% of their time with the novel object, whereas the older animals are showing a much reduced preference for the novel object. But again, the older animals that are treated with the CAW extract are spending more time with the novel object. They're behaving much more similarly to these young animals. And as was the case with the previous test, we have also done this one in our mouse models of Alzheimer's disease, and we saw a comparable improvement with the CAW treatment. So those were pretty exciting behavioral effects, and we wanted to kind of dig more into what kind of cellular changes could be contributing to those effects. And we've landed on 3 big areas where we're seeing impressive effects of CAW: antioxidant response, mitochondrial health, and cerebrovascular reactivity. And I'll just walk you quickly through each of these and show you the data that kind of supports these effects. So starting with antioxidant response. So the endogenous antioxidant response pathway is regulated by this transcription factor NRF2, which under normal circumstances is bound to its chaperone KEAP1 and sequestered in the cytosol. That targets it for ubiquitination and degradation. But if the cell senses oxidative stress, that causes a dissociation between NRF2 and KEAP1. This dissociation can also happen if NRF2 itself is phosphorylated. When NRF2 is released from KEAP1, it can then translocate to the nucleus where it can bind to these antioxidant response elements in the promoter of target genes. And what we've seen in the brains of mice that were treated with CAW is that you get an increase in the expression of NRF2 and its antioxidant target genes. So this is actually data from a different experiment than the behavioral data I showed you before. These mice are slightly older than the mice in the previous experiment, and they were treated with a lower concentration of CAW. But you can appreciate here that there's this coordinate upregulation of NRF2 and these antioxidant target genes in the brains of these animals that were treated with CAW. In this lower panel here, this is actually now looking at a plasma biomarker of oxidative stress, which is 8-hydroxydeoxyguanosine. This is a marker of oxidized DNA damage. And in this case, we did look in the mice that were treated the same way that our behavioral data was from that I just showed you. And these mice, you can see the older mice, this third column over here, have elevated plasma 8-OHDG relative to the younger mice. But the older mice that are treated with CAW, those levels are reduced. And this is really exciting because it's evidence that we have of this antioxidant effect that could be quite translationally relevant since plasma is so accessible in a human population. And then again, we've seen these same kinds of effects in our models of Alzheimer's disease that were treated with CAW. So going on to mitochondrial health, there's kind of 2 parts of this that we were looking at. One was like mitochondrial abundance, and then the other one is more specifically mitochondrial function. And in both cases, we saw a pretty robust effect of Centella asiatica. So this first figure here is, again, looking at gene expression in those slightly older mice that were treated with a slightly lower amount of CAW. And these are genes that encode enzymes that in complex 1, 3, 4, and 5, the electron transport chain. And similar to what we saw in the antioxidant response genes, there's an upregulation across the board of all of these genes. So that might suggest that there is an increased abundance of mitochondria, or it certainly reflects an increased abundance of these particular electron transport chain components. We did see the same effect in our mouse model of Alzheimer's disease. So to try to dig into this a little more, recently we've started branching out into using multiphoton imaging to try to understand how the mitochondria are changing. So this is a pilot project that we've been working on for the last year or so. And this is in our mouse model of Alzheimer's, one of our models. It's the 5X FAD mouse model of beta amyloid accumulation. So these mice overexpress 5 mutant human genes that are associated with hereditary Alzheimer's disease. And this is a very aggressive Alzheimer's model. They start to get these beta amyloid plaques by around like 2 or 3 months. And by 5 or 6 months, they have really robust plaque pathology. And along with that, pretty severe cognitive impairments. You see really elevated levels of oxidative stress and mitochondrial dysfunction in those brains as well. So the way this experiment was designed, on the day that these mice were born, we injected them with a fluorescent AAV marker of neuronal mitochondria. Then when they got to be 5 months of age, we did a surgery to put a cranial window in their heads. And so that essentially is just removing a small piece of the skull and using dental cement, affixing a glass cover slip over top of that. If you do the surgery right, the mice can then go on to live the rest of their lives without major complications. So you can anesthetize them and repeatedly put them under a microscope and see in a live animal over time how some of these parameters are changing in the brain. So for this initial experiment, what we did is we did the surgery at 5 months, let them recover for about a month, and then injected them with a blue dye that stains the amyloid plaques and took an initial image. Then we treated the mice for a month and then did a second imaging session. And this over here on the right is an example of the images we get. You can see the blue are the plaques and the green are the mitochondria. And what we were looking for is if there were changes in mitochondrial abundance in relationship to the plaque, like closer to the plaque or further away from the plaque after CAW treatment. So one thing I want to point out, if you zoom in on this plaque here, you can kind of see that there's a dense core in the center and then like more diffuse halo area around that. And what was really interesting, and I think you can see even in this one example image, is that there seems to be fewer mitochondria close to this plaque and really a lack of them in that halo region relative to further away from the plaque. So one of the first things that we looked at was, does CAW treatment change the abundance of those mitochondria close to the plaque? And specifically, we were looking inside this halo region. And this is extremely preliminary. These are only our first 3 animals, but we did see that there was an increase in the number of mitochondria. And again, you can kind of see here in these 2 images. Unfortunately, it wasn't possible in this initial experiment to find the identical plaque that we saw before so that you could see in the exact same place. So what we ended up doing was taking images of many different plaques throughout the brain beforehand and after and averaging together. But it did seem like there were more mitochondria after the CAW treatment than before. So as I've said several times, this is very preliminary. This study is ongoing and we have a lot more mice now. And so we have enough that we can actually have a group that gets CAW and then a group that won't get CAW. We can compare just the normal progression of the disease, how mitochondria are changing close to the plaques, and how that's altered with the CAW treatment. So this data I thought was pretty cool. It's a cool technique and I'm excited that we're moving forward with it. But it kind of remains to be seen exactly what we'll see once we have a bigger, properly powered experiment. So the other element of mitochondrial health that we wanted to look at was actually mitochondrial function. And we did this using the seahorse platform, which probably you guys have heard of as well. But if you're not familiar with it, it lets you look in either cells in culture or freshly isolated cells or isolated mitochondria at oxygen consumption rates over time as you introduce different inhibitors and accelerators of the electron transport chain, which essentially lets you get this profile of mitochondrial bioenergetics where you're able to tease out a number of different parameters in a single assay. So we did this test in isolated mitochondria. These are cortical mitochondria that we isolated from the brains of those 5XFAD mice that had been treated with CAW. So this is at the end of a month of treatment with CAW. We isolate the mitochondria and run them through the seahorse assay. And what you can see is if you look at this third bar over, this gray bar, those are the 5XFAD control mice relative to the black bar, which are the wild type controls. There is a pretty stark impairment in both basal and maximal respiration that you can see here. But in the animals that were treated with CAW, that impairment is attenuated. So it does seem from this data like CAW might be affecting both mitochondrial protein expression as well as mitochondrial function. We went on to try to look for biomarkers that could be useful of that effect to use in a clinical trial. And this is work that was done in the Benfer Center. It was done by Dr. Ben Zimmerman, who's a very talented postdoc in Anusha Mishra's lab. And what he was doing here is using proton MRS imaging to quantify the ratio of N-acetyl aspartic acid to creatine, which is a marker of like mitochondrial function and neuronal health. And so these mice are treated similarly to the behavioral data I showed you at the beginning of the presentation. They're not the exact same animals. This is a different cohort of animals, but same concentration of CAW for about the same amount of time as well. And what he was able to detect here is that there was a strong trend towards improved NAA to creatine ratio in the hippocampus of these older animals. It didn't quite reach statistical significance, but it was impressive nonetheless that he was able to detect this in these mice. So that was really exciting because, like I said, this is another biomarker that could potentially be used in a clinical trial. So the last area that I wanted to touch on with the potential mechanism here is related to cerebrovascular reactivity. This is another set of experiments that was done by Ben Zimmerman. And again, it's using these imaging techniques that could be very useful in a clinical trial. Same treatments for the mice with the higher dose of CAW for a month. And what he's measuring here by T2 weighted MRI and perfusion mapping is the change in cerebral blood flow following a hypercapnic challenge. So that's like a little transient increase in the amount of CO2 that the mice are breathing. That results in a change in cerebral blood flow. And what he was looking at here is, does that change get altered with CAW treatment in young and old mice? So if you look over here in the graph, you can see if this darker bar, these are the older animals. And if you compare the control to the treatment, the CAW treatment, you can see there's an increase in cerebral blood flow in those animals. And that's what is driving this significant treatment effect that he found in his statistical model. So all of that work was really quite exciting, and we had a lot of good leads for things we could follow up on in an eventual clinical trial. One of the things that we still needed to look at, though, was which are the compounds within that CAW extract that are the most important for the effects. As I've said, this is really critical in terms of creating a standardized product, but also it gives you clues as to which compounds you can be trying to quantify in like the blood of patients to make sure that you're getting high enough levels that are to what you were shooting for when you picked your doses. And these two compound groups that I have circled here, they really came to our attention based on previous work that we had done, including some studies that Doris's lab did in Drosophila, and we also had some cell culture data that suggested that these triterpenes and caffeoquinic acids, or CQAs, are really important for the effects of CAW on locomotion in the Drosophila and also dendritic arborization in primary neurons. So just to zoom in a little more on these compounds, the triterpenes, there's 4 of them that are kind of the signature compounds of Centella asiatica. Asiatic acid, madecassic acid, and then their glycoside versions as well. And then there are a number of mono and dicaffeoquinic acids that are found in CAW. And these compounds, these CQAs, are not at all unique to Centella. They're actually found in a huge number of different plants. So we were maybe at first a little surprised that these ones had such a important effect, considering that this plant in particular is used for cognitive enhancement and these are not unique to it. But it does seem like there's something about the combination of them that's really necessary for evoking these effects. So based on this data that we had from, like I said, Doris's lab and some other work that was done previously, we decided to evaluate the effects of these different compound groups in our mouse model of aging. So in this experiment, we didn't have any young mice. It was just 18-month-old mice, and they were either treated with a vehicle control, the triterpenes, the caffeoquinic acids, or triterpenes and caffeoquinic acids together. And the concentrations that were used in each of these groups was proportional to their relative abundance in 10 grams per liter CAW. So that's the concentration that was given to those mice that for the behavioral data I showed at the very beginning. Because ultimately our goal was to compare the behavioral response to these ones to what we saw with the whole extract to get a sense of which compounds are most important for that effect. So it was the same kind of paradigm where in the last 2 weeks they underwent the behavioral testing. And what we saw was pretty interesting. This is the first phase of that odor discrimination reversal learning test, the acquisition phase that specifically looks at learning. And remember, in this experiment, lower bars indicate better performance. And what you can see is relative to vehicle, there's an improvement in performance with all 3 of the treatment conditions. But in terms of the magnitude of this difference, it's even more significantly different between the TT and CQA versus vehicle than it is with the TT plus CQA, which is interesting. Looking at the cognitive flexibility part of the test, which is the shift phase, we saw a similar pattern. Here you're seeing the data split out by each sex separately, whereas previously all the data that I showed you had both sexes combined together. And that's because when we did our statistical analysis for all of the previous data, we never saw an interaction between sex and any of the endpoints we looked at. But in this phase of the test, we did see a significant interaction between sex and response. And you can see why that's the case. If you look here at the females, you're seeing a much more robust effect of especially the CQA and TT plus CQA conditions that you're just not seeing in the males. But overall, this looks quite similar to what we saw with the acquisition, that you're getting an improvement with all of these conditions. The real question, though, is how does this stack up against what we saw with the entire CAW extract? So to do that, what I have plotted here is the data from that original experiment against the data from this experiment. But to account for the fact that they weren't all together in the same experiment, what's being represented is the fold change relative to the control in their respective experiments. So this CAW value here of 0.6 means that there was about a 40% improvement in performance relative to controls with CAW, whereas for the TT, that's more like a 30% improvement, and this is maybe 20, and this one more like 10. And looking at it this way, it's really clear to see kind of the stepwise comparisons here, and the overall message that even though you're getting improvements with the TTs and CQAs separately, and some improvement with them together, it's not the same degree of improvement that you get when you give the entire extract. And it's quite similar when you look in the shift phase, which again is about the part that gets at cognitive flexibility. Same pattern as we saw with the acquisition phase, that although there is improvement with the compound groups themselves, it's not as robust as you get with the entire CW extract. Looking at the NORT data, we saw the same kind of pattern. In this test, higher bars are better performance, and you can see in the experiment that we did with the compounds that there was a trend towards an improvement with the TT condition, but it wasn't significant with, I mean, it wasn't significant with the TTs, but there wasn't even a trend with the CQA or TT plus CQA. And then again, plotting it against on the same axes as the CAW experiment, you can really see how the TTs are relatively comparable, but the CQA and TT plus CQA groups are just not evoking the same benefit. So this was important data to have as we're thinking through to a clinical trial, because it seems pretty clear that the TTs and CQAs are contributing to this effect, but they can't fully explain the effect. That there's other compounds within the extract that are important for getting the maximal benefit. And it was also really interesting to observe in these different tests that the combination of TT plus CQA is not performing as well as either the TTs or CQAs separately. And that was something that Doris had seen in her experiments as well, which again just kind of points to other compounds within the whole CAW extract that are preventing whatever inhibitory effects are happening when you are just giving all of the TTs and CQAs without the rest of the CAW extract present. So moving forward to clinical trials, we kind of had settled on we're going to need to use the entire extract because we can't point to one compound group that's driving all of the effects. But we do want to kind of track the triterpenes and CQAs, both in terms of their abundance in the preparation that we're going to give, and then also in the blood of patients going forward. So based on all of this preclinical data that we generated in BENFRA and in some other studies related to BENFRA, we felt like we were ready to start a clinical trial. And as I said, this fall we completed a early stage safety and target engagement trial. So we're definitely still not at the efficacy stage, but this was pretty exciting to see all the work that we had done in the preclinical space actually moving into the clinic. So this is just a little roadmap that the National Center for Complementary Integrative Health put out on how to design the maximally effective efficacy trials for natural products and botanical agents. And looking at this, we realized that the steps that we really needed to fill in before we could start our trial was thinking about dose selection, product development, pharmacokinetics, and then safety and target engagement. All of those had to be done before we can do an efficacy trial. So starting with dose selection, we had all this data in the mice showing that there were improvements in cognitive function when the mice were given a range of doses of CAW between 2 and 10 grams per day. So using interspecies scaling, we were able to calculate what that would be for a human, and then cross-referencing that with published reports of human doses of either preparations of Centella or even the isolated compounds, we settled on using 2 and 4 grams of CAW per day. The next hurdle was product development. 2 and 4 grams of CAW powder ended up meaning something between like, I think it was 8 and 16 capsules per day if we were going to encapsulate it, which was just completely not feasible for a clinical trial. that's a huge number of pills for anyone to take. And especially since our target population is cognitively impaired, it was just unrealistic to expect them to consume that many capsules in a day. So we ended up focusing on creating a drink mix that could deliver the CAW instead. And this work was spearheaded by Dr. Kirsten Wright. She really did a ton of work on the product development of this, and she also did the PK work, which I'll show you in a second. But she was able to find excipients that did a great job of placebo matching. They had relatively similar palatability to the CAW, no safety issues, and importantly, they didn't contain any TTs or CQAs. And this was actually a really hard step for her to figure out because TTs, not so much TTs, but the CQAs are so abundant across different plant materials that it was very hard for her to find the right excipients that didn't contain any of those. But she did, and this is a picture here, and you can see that she did a really great job of getting it to match, at least visually, between the different concentrations of CAW. So she then went on to do this PK study. The product that she has now is the Centella asiatica product, so it's capped instead of CAW, which I was talking about before, but it's also our CAW extract just prepared in that drink mix. And she was evaluating it in older adults with MCI and found that there was a nice detectable difference between the two doses, looking at these two triterpene compounds. And when she went on to look at other metabolites and other compounds within the CAW extract, for the most part, she also saw a measurable increase with the higher dose compared to the lower dose. So that was really great. That was a major hurdle to move us towards our first safety and target engagement study. And that's the one that we actually just completed this fall. So we're still moving forward towards an efficacy trial, and we're not there yet, but I do want to tell you about this safety and target engagement study that we did. I guess I should lead by saying it just ended in October. So we're still blinded about which group is which, but I'll show you what results we have so far and where we could see differences between the groups, although we don't know which group got Centella and which group got placebo. But the population we were studying were elderly participants with MCI or mild Alzheimer's disease. We had 32 total, and we were aiming for an even split between the sexes. I'll show you the consort diagram in a second. We didn't quite get that, but that was the goal. And they were given this Centella at 4 grams or a placebo for 6 weeks. And this is randomized, double blind. And the primary endpoints are biological signature of target engagement and safety and tolerability. So the signature that we are looking at is this 8-OHDG, that marker of oxidative stress. We looked both in urine and plasma. And then we had some of these imaging parameters that we were looking at as well to look at mitochondrial activity and neuronal health. Then we had a lot of safety and tolerability endpoints, specifically questionnaires about adverse events, but also routine blood and urine tests that indicate safety. Beyond that, there were a number of exploratory metrics that we were looking at, which included expression of that antioxidant transcription factor NRF2 and looking at mitochondrial activity in peripheral blood mononuclear cells from these participants. We also looked at plasma levels of some of these kind of classic AD biomarkers, as well as plasma levels of those signature triterpene compounds. And then we had some surveys of cognitive measures as well. But the study wasn't really powered to look at any of these endpoints. These were just extra ones that we were hoping that we might see an effect in. The primary endpoints were this biological signature and then safety and tolerability. So the way the study was designed, there was a screening visit, which included cognitive testing and blood and urine tests. Once they passed the screen and were enrolled in the trial, they came in for their first visit, which involved 2 MRI scans, more blood and urine tests, and then several questionnaires. Then they were randomized to either CAW or placebo. The CAW, remember, is 4 grams given as a little like sachet of the powder that they would resuspend in water, or a placebo, which they did 1 per day for 6 weeks. Every 2 weeks, they got a phone call to track symptoms. And then at the end of the 6 weeks, they came back and did all the same testing that they had done initially. So again, the MRIs and blood and urine tests and questionnaires. Then treatment ended and they were followed for 4 additional weeks. Beyond that, just monitoring symptoms by a phone call. So this is the consort diagram. Like I said, we're still blinded. So they're called Group A and Group B, and we don't know which is Centella and which is placebo, but they ended up with pretty comparable numbers, 18 in A, 19 in B, started out. There was a pretty comparable level of people who discontinued the intervention between each of them. In both cases, in Group A, there were 3 that didn't discontinued, and in B, there were 2. It was 1 in each group was because of adverse events. And then in the Group A, there were 2 people who couldn't tolerate the MRI. And in Group B, there was a person who had an unrelated surgery and had to withdraw. So we ended up with 15 in Group A, 17 in Group B. And the male to female split wasn't quite exactly even like we wanted, but it was pretty close. So these are our preliminary results, and I do want to stress that these are unpublished, so please don't share them with anybody, but there's still quite a bit that's being analyzed. We definitely don't have everything analyzed yet, which is why we're still blinded, but there are some interesting effects that we're seeing. There is a significant difference between the groups in ATP levels based on our imaging techniques. And there was a trend towards differences in plasma NFL, which is neurofilament light. It's a marker of neurodegeneration. Not quite significant, but a trend towards a difference between the groups there as well. Importantly for this study, there was no difference in the emergence of adverse events between the 2 groups, and there were no detrimental changes in the blood and urine safety labs for either group. So that's great. That was like a major endpoint of the study. This ATP data and NFL data is encouraging, but we were a little bit disappointed that we weren't seeing more of an effect of our biological signature. We didn't see a change in urinary 8-OHDG, although we still haven't analyzed the plasma levels yet, and that's where we saw the difference in the mice. So it's possible we will still see an effect there. We weren't seeing much of a change in the PBMC NRF2 expression or mitochondrial activity, and then there weren't any changes between the groups in the Alzheimer's disease biomarkers. So to summarize, what I showed you today is a lot of mouse data from models of aging and Alzheimer's disease, showing that this water extract of CAW has pretty impressive effects on learning, memory, and executive function. It does seem to improve antioxidant response, mitochondrial health, and also it improves cerebrovascular reactivity. The TT and CQA compounds definitely seem to contribute to the effects, these cognitive enhancing effects, but there are likely other compounds within the CAW extract that are also important for that. So that's an area of ongoing work for our center. And then finally, we're really close to having the data from this first safety and target engagement trial of CAW. And that'll be super exciting to see how that shakes out. And hopefully we can continue on from there to move into some actual efficacy trials soon. So with that, I'll stop. This is just the huge amount of funding that went into all of this work. Specifically the mouse work that I showed, these are the people who were very involved in that. And then these are all the huge number of people who are involved in that clinical trial, because it really takes a tremendous amount of people. And then I'll just end with this one, which is all of our Benford team. It was a huge collaborative effort. the center, the U19 center, was funded through 2025, and we're in a no-cost extension till the middle of this year. But we're actively seeking out more funding opportunities because we really want to continue doing this work. It was just so much fun to be on a team and get to see such a wide range of research all kind of moving towards the same ultimate goal. So stay tuned. Hopefully we'll have more coming out of the Benford Center in the future. But that's all I had for today. So if there's any questions, I'm happy to try to answer them.
So if there's a question. Stephen?
Thanks, Nora. That was really outstanding. Had a question whether or not you've had some medicinal chemists or folks that are really following what would be blood-brain barrier permeable. When I look at the triterpenes, they look enormous relative to being able to get past a blood-brain barrier. And I was just wondering whether there's any indication that some of the other molecules might be transiently changing blood-brain barrier permeability that would allow for that to be able to get in and have an effect. But I just didn't know, it could be, the glance at the triterpenes, maybe for some reason they are potentially permeable to that barrier.
Yeah, that's a fantastic question. And you're right, there are huge compounds. It's hard to imagine that they're just going to get in on their own. Um, that's something that we're looking at pretty intensely in our group. First of all, seeing if we can detect any of the compounds in the brain, because if we can, that might suggest what you were talking about, that maybe there's an effect on blood-brain barrier permeability. Some of the work that Anusha Mishra is doing in her lab, some of her imaging techniques can be used to kind of gauge permeability. So that's something that I know that Ben is interested in kind of seeing if he can get any additional information from his imaging data that he has. But we don't have any direct evidence right now that there's a change in blood-brain barrier permeability. But I would say that we haven't definitively shown it either way. I mean, another possibility is that they don't directly get in the brain at all, that it's a peripheral effect that is changing something else that gets into the brain. Like, it doesn't necessarily have to be a direct effect in the brain, but we don't even know that much yet either. Like, that's probably the biggest area that we still need to delve more into, is like what is actually what is the brain exposed to, and how, if it is getting in, is that happening?
Are there any indications that there are changes in the relative levels of inflammation? Um, uh, Whether or not this could be potentially modulating various responses from that perspective, and therefore translating relative to effects in the brain. And the other question I had was, when you're measuring the 8-hydroxyguanine, is that the free base or is the base still with its sugar? Because that would, if it's the free base, then there could be an indication of the DNA repair processes which come in and lop out the
Right.
8-hydroxyguanine versus degrading DNA where you would generate the nucleotide.
Yeah, that's such a good question. I'm trying to remember the exact kit we used. I want to say that it was not the freebase, but I would have to double check to confirm that for sure. In terms of your question about neuroinflammation, there's actually a lot of literature about anti-inflammatory effects of Centella asiatica. We haven't looked into that too much in our mice. Um, Although one of the postdocs, he's now an assistant professor, Steve Chamberlain, he is in the middle of running another experiment in aged mice with Centella asiatica, and he was looking at CRP levels in the plasma, and I'm pretty sure that his data is showing that those are reduced with the Centella treatment, but it's definitely not something that we've looked into to any great degree. So that certainly could also be another relevant mechanism for sure.
So there
Thank you
was
very much.
a question in the chat, and I guess I can read it. As there are promising results for mitochondrial health and ATP, is there any future plans to measure effect on physical fitness metrics?
Huh. Yeah, we don't have any plans for that right now, but that would certainly be interesting. And actually, the study that Steve is doing right now, he's looking at the effects of Centella and exercise and the combination of them together in old mice to see if there's any kind of synergy that's happening there. So that doesn't specifically get at your question about physical fitness, but he is measuring a lot more motor functions and markers of cardiovascular health and that kind of thing in those animals. So maybe we'll have some answers, at least some clues to that in the future. But as far as I know, that's the only thing that's even a little bit related to that. But it is a really interesting area.
Any more questions? I have actually one which I thought about. So I mean, you didn't get too many effects, too much of an effect on the measurements in with the clinical trial. So the mice were treated for 2 weeks, right? And the humans got it for 6 weeks. Is that comparable if you compare it correlated to the lifespan? Probably not, right?
Probably not. Yeah. The mice were actually treated even longer, 2 weeks before the first day of the first
The test was,
behavioral test. So all the changes we were looking at in the brain, at least, were after 4 or 5 weeks of treatment, which is an even greater percentage of their life, like you're talking about. Yeah, obviously you're limited in human research with like the costs associated with doing those super long trials, but I think that could certainly be a contributing factor to why there wasn't a more robust effect.
Yeah.